Thank you very much, and good afternoon to everyone in the audience. I am the leader of this project. I am going to introduce the main concept behind the project and some results. The project is towards the end. So Simtap stands for Self-Sufficient Integrated Multi-Traffic Aquaponics System for improving food production sustainability and brackish water use and recycling. The project was approved at the end of 2018 in the framework of the first PRIMA call. PRIMA is the Partnership for Research, for Innovation and Research in the Mediterranean area. The project is carried out by 8 different entities shown in this slide, in which 6 academic or scientific institutions are supported by an high school in France and a consultant from Germany. Not going? Okay, thank you. So the slide shows the main goals and the work plan of the project. The idea of the project is to test the performance of 4 Simtap prototypes built in France, Italy, Turkey, and quite recently in Malta, and to identify the main drawbacks and obstacles to the application of the Simtap concept to saltwater aquaponics. And of course, one important goal is to assess the sustainability of this system through LCA, LCC, and EA analysis. You see in, I have to close this. Thank you. And the project consists of different work packages, and one of which is devoted to the life cycle assessment. And this workshop was nicely organized by Jacopo Bascianetti within the work package number 5. And the responsible for this work package is Joel Obin, who is among the speakers today. What are the expected benefits of the Simtap? You know that, and we saw that in the first presentation this afternoon, that aquaculture depends on commercial fish feed that is based on the fish meal, fish oil, cereals, and soybean. So they depend on fishing of forage fish and intensive crop system. So they have a very large environmental impact. Moreover, especially in land aquaculture systems, there is a huge use of water and contamination as well. And finally, an important goal of the project is also to try to exploit greenhouse effluents to provide some benefits to the aquaculture system itself. As such, the Simtap system is an alternative technology to the RAS, or recirculating aquaculture system, or the flow-through aquaculture system. The main principle of Simtap is that some organisms grow in the system itself, namely polychaetes, worm, mussel, and clams, which are the detritivores and the filter feed organisms, or DFFOs, from now on, can replace, at least partially, the commercial feed, thus reducing the environmental impact of the aquaculture production. The Simtap concept was applied in 2 different types of prototype. One system was the prototype built in France, and which is described in this slide. In this system, different species at different trophic levels are grown in ponds with an input which is represented by feed not containing fish meal and fish oil, which in part consist of mussel and vegetable discarded from the local market. This system is the system used by the French team in the project. The team leader, as I said before, is Joël Aubin, and for sure Joël is ready to explain and to provide you more details and information on the work and the system they are using in France. The slide shows some picture and some results of the activity conducted in France in the project. In Italy and also in Turkey and Malta, the prototype, the Simtap prototype, is based on a different concept. Actually, the Simtap in Italy, for instance, the system I am going to describe now, is a coupled aquaponic system with some important modification, however. You know that in aquaponics, crop plants and fish or other aquatic species are growing the same water loop. In this system, there are two main inputs: feed for fish and fresh water to compensate the water loss due to evapotranspiration. There are also important outputs, of course, fish and crops, but also sludge, which originate from the mechanical filtration of the solid waste produced by the fish. In the Simtap, the mechanical filtration is replaced by DFF organisms, I mean polychaetes worm, mussel, and so on, which are cultured in the system for phytodepletion of the water exiting from the fish tank and can be used to feed the fish. Of course, in a system like that, the production of these organisms is not enough to feed the fish. So we need another feed source for these organisms. In the system, these organisms are microalgae. Microalgae are good feed, valuable feed for polychaetes worm, for instance. They provide the lipids, protein, vitamin, and so on. Of course, microalgae need some nutrients, and so microalgae grow in low-cost photobioreactors on the effluents from greenhouse hydroponics, which are still rich in nitrogen, phosphorus, and so on, and so these effluents must be treated before discharging into the environment. That's the concept behind the Simtap prototype built in Italy, but also in Turkey and Malta, as I said before. Of course, Simtap deals with saltwater system. This means that the crop plants must be very tolerant to salinity. And this is because in the Simtap system, halophytes or seaweed like ulva, for instance, are grown together with fish like sea bream or sea bass. This is a sketch of the prototype in Italy, built in 2020, which hosted in the last 3 years a number of experiments with fish, with some allophytic species, allophytic plants, a native polychaete worm species, Ediste diversicolor, we captured it in a river close to Pisa, on the microbial community in the biofilter, and also on Ulva rigida, the seaweed we grow in our system. You can see some picture of our system there. The salinity levels tested in Pisa were 25 grams per liter of PPT in 2021, and much lower salinity this year with another species of fish. The fish species studied in the project are shown in this slide. sea bass and sea bream. And about the crops, the most interesting plants seems Salicornia europea, also called the glasswort or sea asparagus. It's an interesting species because there is, in some countries like France or Israel or Morocco, for instance, there is already a significant market. In Italy, to be honest, the market is still potential. And these species can be grown successfully, at least in spring, summer season. And because the retail price is quite high, as in some period it's even higher than the retail price of the fish you can see in this slide. A slide dedicated to Salicornia europea. We found that these plants can grow at very different level of salinity, up to 30 ppt or gram per liter, which is a salinity very close to the seawater salinity, which is 35 to 40 ppt. Actually, Salicornia need saline water in order to produce succulent and greeny shoots as the market demands to this kind of plants. Just a result of the experiment with the sea bream in 2021 at 25 degree gram per liter, of salinity. And in this experiment, we tested two different diets based on the commercial feed containing fish meal and fish oil against an alternative feed consisting of a mixture of polychaete worms, mussel, and clams only. You see that we were able to reach the market size of the fish in less than 10 months, and with no important, no significant differences among the two treatments, the two different diets. And you see also on the right column the final density in different stage of the growth, from more or less the beginning of March till the mid of December. And in the table on the right of the slide, you see that the growth performance we found in sea bream in Pisa is more or less the same of the growth performance we found in the literature in fish grow in different systems, from recirculating aquaculture system and in sea gauges. After 4 years of work with the prototype in the laboratory, many empirical observations, some mistakes, to be honest, a couple of accidents, and almost daily discussion among us in Pisa, I think we can say what went right and what went wrong. As I show, apparently fish can reach market size in less than 1 year with an alternative feed consisting of only polychaete worm, mussel, and clams. We also found that the native polychaete worm, Ediste diversicolor, show a good adaptation to the salinity of the Simtap, to the high salinity we tested in the Simtap prototype. And also Salicornia proved to be a good candidate crop. However, it grew very well in summer, spring, but much more slowly in winter. This is a problem. And the culture of mussel and clams was completely unsuccessful, probably because the low salinity, the relatively low salinity. Reducing the salinity, the water salinity, say lower than 5 ppt, may increase for sure the list of candidate crops. On the other hand, we found that low salinity can make more difficult the maintenance of a high-quality water environment in a system without mechanical filtration. Because don't forget that in our system, mechanical filtration is replaced by the presence of detritivore and the filter-feed organism. That is an important point. And moving quickly to the conclusion, I think I am right. Thank you. We, towards the end of the project, we have changed our idea of the Simtap. At the beginning, we talked and we built the prototype according to the principle of an completely coupled aquaponics system. And we feel that after some experiments and towards the end of the project, we think that a more efficient system is an decoupled layout where the water loop for the fish is separated from the water loop for the hydroponic cultivation of crop plants. And we also think that cannot replace mechanical filtration, rearing some organism in the system itself. And this means that the sludge produced by the fish must be treated in a different way. We are just thinking about vermicomposting, for instance. However, we have to solve the problem of the salinity of the waste material produced in the system. Thank you very much, and thank you.
Thank you very much. If there is any question, either in the audience or online. Okay, we have one question online. Is adding species to the system increasing risks or creating resilience?
Difficult question. The main problem is with the intermediate organism. So we were able to rear only worms, but we need probably we need a different species of worm with respect to the native species we used to rear in the last 2 years, because the biomass is not enough for efficient biofiltration of the recirculating water and to provide enough feed to the fish.
All right. Thank you very much.
Thank you.